Nellie was so wonderfully kind. So we are now left with 17 minutes to the end of our session. So I'm afraid we all have talked a little bit long, and it's probably, even though I hope, oh, there it goes. I hope that I can go very quickly trying to give you some of the basis of the heterogeneity within the microenvironment and why context is so important. And you have to forgive me, many of you have heard me, and for those of you who haven't heard me, I'm very happy to provide you with the papers. I'm old enough, really old enough. I have been around for 35, 40 years, basically step by step trying to show some of the concepts. And today I changed my talk 10 times because I would listen and I would just say, oi, we have to do something here. So, you all start with that many trillion cells. And these trillions and trillions of cells have the same genome. So think about the idea that when we look at ourselves in the mirror, I look at you, you're all a miracle, because they have the same genome. So if the genome was dominant, you would be a lump. And not that genetics is not important. Your mom and dad met and you are who you are. But once you're born, you're not a lump, and something else determines your nose and your finger and your breast and whatever. And what is that something? And this how is in the heart of understanding cancer. When cells or tissues don't respond to what initially made them you. So if we are to understand cancer, we got to understand the normal tissues in which these cancers arise. So we chose the mammary gland for many different reasons. They change again and again. And the point here was, can we make the tumor cells to remember and say, hey, I am a breast? I mean, after all, they came from the breast, and there is no reason why they couldn't turn into something else if, in fact, they had the right signals. So the mammary gland has many of these things. And I see that I don't have a pointer. I have my thing in here. OK. So yeah, I'm supposed to actually do this here, but I'm not used to doing it, so please forgive me. I'm old-fashioned. So here it is. So there are many of these things, as you know, in a breast, many different parts of the breast. We have 5 or 6 of these. And the idea for me that had a very different training than most people in this room was that I wanted to understand what's the basis of tissue specificity. And I didn't start answering something important about breast cancer, but we decided that even putting the cells and thinking about them and thinking about the mammary gland, as much as it was plastic, we really had so many different components in it. So we settled on this, and we said, oh, this is simple. It has one layer of luminal epithelial cells, one layer of myoepithelial cells, and then this other thing around it. And to make a very long story short, clearly when we wanted to do this in culture, these cells completely forgot where they came from. So there was no essential cell autonomous behavior. They were making gobs and gobs of milk in the mouse. We put them in a dish. We give them all the hormones. They completely forget, and I wish I had time to show you how they look. They forgot to make milk, whatever. So we began to think about, why on earth do they do this? They were making these huge amount of milk. They look so beautiful. Why do they look like this? And we decided that these molecules that are around them, meaning the extracellular matrix molecule, huge insoluble molecules, had to have information and had to tell the cells what to do. And at that time, everything that signaled was soluble. So it was even before integrin. So I postulated that the outside of the cell has to tell through the receptor to this extracellular matrix molecule, had to talk to cytoskeleton, had to talk to chromatin, and this interaction to get your 70 trillion cells to do the right thing, they all had to be dynamic and reciprocal. and they have to be regulated by their particular microenvironment. So in those days, I used to make that in the lab, and now I wish I had patented it, but anyway, you can put these things on top of this matrigel, and in no time at all, here is what we can do in culture, here is in vivo, and some of you are sick of having seen this, but we make these beautiful things, and many of them are full of milk. They are gorgeous, they are polar, they have all the right components, and they say, hey, I'm a breast cells once I'm in 3 dimension. You put them in 2 dimension, they don't do that. So yeah, we can do this with mice, we can do it with humans. I have done a lot with Ole Peterson. And we just said if the normal mammary cells can remember what they are supposed to do when you put them in 3D, maybe the tumor cells think they are tumors and they won't do it. And all of a sudden, we have a gorgeous assay for finding out what is normal, what is malignant. We did these experiments, and here are, as you see in this assay, every single cell here makes a beautiful 3-dimensional structure. Every tumor cell, malignant cells, make a beautiful tumor. So rather than injecting everything to the mouse and waiting 6 months and seeing if this got tumor or not, we can do this now these days in 4 days. And we can find the difference between normal and malignant, and these are all clonal. So every one of those are not single cells, they are those. And these things, every one of them can be taken out, and you can analyze each of them separately. They are like hundreds and thousands of human tumors in one little dish, and the same with hundreds and thousands of normal acinon. Okay, now I didn't want to compare and say, how do these cells know and how come these cells don't? And we looked at a model that Ole Peterson and his former professor had developed, and it took us about 20 years, but it went from reduction mammoplasty of a cell in a tumor all the way in culture under very defined condition, going from non-malignant, coming from reduction mammoplasty to malignant. So it's the first human model that is outside human becoming malignant. But if you look at these cells, they all look the same. And if you put them in 3D, in 3 dimension, all of a sudden they sort out. And they sort out on passage 60. There is a lot of amplification and deletion. But these things have lost P53 and whatever, but they still say, hey, I'm normal. In 110, they are MYC amplified and God knows what else. They still think they're normal. We're actually in passage quite way out back there, and they still think they're normal. And in fact, we have clones that act like this and not like that. However, when you look at the cells that were injected into the mouse, and once in a blue moon they became malignant, eventually, they are pre-malignant cells. You see that how much mutation they have, and you see that the structure goes before cells become malignant. That's a really important thinking. The structure goes before the cells become malignant. And this was based on these cells, and if you look at this, we have so much data about these cells. We have also a whole lot of additional cohort in the middle. Okay. So here is the non-malignant, here is the malignant. Those normal cells had a lot of mutation, and they still thought they were normal. So we thought, well, on the basis of this, and on the basis of other things that maybe I have time to show you one slide, it's completely clear to me that the structure was dominant. So we said, if this is the case, why we don't take cells like this and actually make them think they are normal, if I'm right, and restore the structure, and we did that. And we measured the stuff. Valerie Weaver, a very brilliant fellow in the lab, who now has her own lab at UCSF, I should say her own empire. And these cells, they had many different kind of receptors, and these had it too, but the ratios were completely different. The balance of signaling was gone. And so we took beta-1 integrin Irv Weissman talked about that. We discovered that many years ago, actually towards the early 90s, that this molecule was extremely important. So we used the inhibitory antibody to beta-1, and it does exactly what Irv Weissman was talking about his molecule. So when we did that, look what happened. The cells did this. They're not completely normal. They are making this gorgeous structure. They are completely stopped growing. And we now know a lot about why they stop growing, because when you add laminin, there is actin in the nucleus, and the laminin is talking to actin, and when actin exits, you add the laminin, actin exits, and the cells become quiescent, just like dormant cells, and every one of them does it. So we didn't select, we didn't do anything. 10 to the 7th cell revert, and we have many movies of these. They all look, and people still wouldn't believe it, so we had to show that it was reversible. And here are the non-malignant, here are the malignant cells without the antibody. Here are the malignant cells with the antibody. And now if we take these cells, put them in a dish, and then take the antibody away, it becomes like that. If we add antibodies like this, and of course, what does that tell you? We can do it over and over again, that even in malignancy, phenotype is dominant over genotype. Now, it doesn't mean that I can take a tumor and revert it, okay? So don't get confused about that. I don't have a whole hour to tell you about all of this. But here was the interesting thing. We got the beta-1 integrin. Why did the cells stop? They had 6 times the level of EGFR. Why did they stop? Well, in 3 dimensions, the down modulation of beta-1 down modulates EGFR. So we measured EGFR. EGFR was 1x, not to 0, 1x. This became 1x. And then we looked at all of these things. Every one of those gets completely reverted. Cells talk to each other when they are in the right context. And they talk to each other in a way that they integrate all of the signaling molecules in order for the tissues to be able to do what they do. And what is dominant is the architecture of that tissue. So as I said, we could even do metastatic cells, so form and function are related. We said, if this is correct due to some stuff I had done in the 70s, long before any of you were born, the question was, what about glucose, if everything has to integrate? What about glucose metabolism, which now has gotten famous again? In the old days, people thought I was nuts doing glucose when everybody was doing oncogene. Okay, so we have done a series of experiments, came out in Journal of Clinical Investigation just in 2014. And it took a long time because the reviewers simply couldn't understand why it would do this. So the long and short of it is, here are non-malignant cells. You see they make these beautiful structures. Now we take the malignant cells and we put them in 0 glucose. Look at them. Identical. They revert, they become normal looking. We have done so much biochemistry. We know the entire pathway. We have new pathways. It's not through HIF as it is in 2D. And what happens, these cells have 400 times of a glucose transporter. We take the glucose away, it goes bang. Within 6 hours, it becomes 1x, just like the non-malignant cells. And as you increase the glucose concentration, you see that they become a mess. So don't have your second cheesecake, please. Glucose is not good for you. Okay, so we put glucose here, and you see that that is one of the things that changes. So these questions, what then is oncogene? What does oncogene addiction mean? Indeed, all of it doesn't make sense to me. I think they really cause trouble in our brain. This is an oncogene, this is an oncogene, this is an oncogene, or whatever, if you like to talk about it, and they all talk to each other within the tissue, even within the malignant cells. This is a proof of principle, okay? We can do it with many different cell types. So what mutation is causal, or which is the driver? That's why I was getting up, giving everybody a little bit of a hard time. So growth and malignant behavior are regulated at the level of tissue architecture. And if that weren't the case, again, I rest my case that you would not be who you are. This is what has to happen. So if you're not convinced, I give you something else I did in the 80s. So this is Rat sarcoma virus, the most important potent oncogene ever. And there are still people today who say single oncogene is enough. Now, please believe me, I'm a believer in genes. I'm not saying anything opposite that, as you will see. So here is this ugly tumor. 1911 got the Nobel Prize 50 years later. People forgot about the tumor because they had the virus. They could add it to the cell, and they would say, oh, this bump is malignant, this bump is malignant, this one is normal. And I had no idea what they were talking about. What's normal, what's malignant in 2 dimensions? So we injected the virus into the chicken embryo. It integrated. We published it in 1984 in Nature. No tumors. No tumors. Nobody believed it. So many years later, we attached PP60 sarcs to that lax Z. Sorry, I show the same thing because I think these are so black and white. I mean, look at those blue cells. Every one of them could be a tumor, but they are this beautiful feather. Now, if we took that feather and dissociated it and put it in a dish, overnight, we get a pile of blue cells and we get malignancy. And why the embryo does it and and chicken doesn't do it is a gorgeous study. Hardly ever anybody asks until they go home and say, why? And then they call me. So you're welcome to do that. I'll tell you when it comes. Does context, i.e. the microenvironment, determines what even a potent oncogene can do, but how does context signal? Now here is a disclaimer. I do believe in genes. Please, people have accused me of this. Cancer, I was a bacterial geneticist. And before that, I was a chemist. So I had the advantage of not getting corrupted by the textbooks. So I just looked at the data and said, this is what's happening. I love your questions. Okay, so by no means those mutations are enough. If you had 1 or 2 or 5 mutations and you got tumor, folks, you have 70 trillion cells. Can you imagine how many of those, even if they are 0.00001, is an oncogene? Again, you will be a mess. Okay, so this is a hopeful view of cancer. It means that we should be able to take the solid tumors and treat them like some kind of lymphoma, where people actually give retinoic acid or they give whatever else, and they can make the tumor sort of stabilize. And I think that it's not going to be a simple idea because we have to treat the tumor and we have to treat the microenvironment. But here is another fantastic example of this. What kills the patient is not the primary tumors. You can do as many mutations as you like. What kills them is the dormant tumors that goes and somewhere sits there. This paper we did in Nature Cell Biology by Cyrus Qajar, a fantastically brilliant postdoc. Here, I have again only a few minutes to just show you this, but here is a three-layered model we made to model dormancy of a cell line that was, or a breast cancer cell that was not metastatic. We injected it into the heart of the mice, and these cells removed the primary tumors, and then the cells went to brain and bone and liver, you know, all those places, or lung, all the places they have to go. And when Cyrus looked, they were sitting on the blood vessel. The cells that had become dormant were sitting on the blood vessel. So we modeled it. This is the same tumor within the same plate. Here are stable endothelial cells making these gorgeous looking things over a niche of either lung or a hematopoietic system. When they are on a stable blood vessel, they're sitting there without growing. However, in the tip of these blood vessels, as they start developing, the same cells, if they're sitting here, they make a huge tumor, and they're local. They're as local as they come. So it just shows you how microenvironment of this is different than that, even though they are the same cells in the same dish. They are being, in fact, never mind that. Where is it, this thing? Yeah, okay. So we did mass spec. We know what this, I think, was on the cover. If you look here, you find out what are the molecules that are involved here, what are the molecules that are involved here. Why is laminin important? I already told you, if you add laminin, the cells become quiescent in 3D or even in 2D. And that's because laminin is directing the chromatin through a very complicated and lovely program that I don't have time to tell you. But if I have to tell you one thing, a paper that is under review is we show there's 7,000 papers about p53. And you say to yourself, well, if the extracellular matrix is important and is regulating, what should happen? There should be some connection between the two. And sure enough, what regulates p53 is laminin 111. And what regulates laminin 111 is p53. It's dynamic, reciprocal. p53 binds to the promoter. It's so pretty. I wish I had time to show you. OK, so I want to say another thing about cancer. This guy, this guy in the lab, dad of two, has spent years of showing that the breast, in fact, is not just luminal cells and myoepithelial cells. There are 12 different cell types in the breast. And Carlos Cardas, I was just at Heidelberg giving them this, one of their keynote talks. I had no idea what I was doing there because it was a stem cell meeting, but here I was. But he got up and showed that it will come out in Nature, that he can actually define 10 different kind of breast tumors, not 4, but 10. And he was very interested that we can find 12 fractions of these. And so I want to quickly summarize and say what is important is not mutation alone, but for sure mutation is important. I have never seen a tumor that doesn't have mutation. Those people who use my name or my work and say, yeah, but we don't need mutation are, insofar as I'm concerned, are completely wrong because we don't have the data. We don't have any data to show that mutation is not important. Of course it's important. So what is important is not mutation alone, biochemical signaling alone, force and tension alone. We have all of that too. It is the integration of chemical, mechanical, physical, biological, and as yet unknown principle that makes us who we are. And I am now out of time, but here lies the problem with cancer, because it's such exquisite balance we have developed throughout the evolution, and now this thing goes off, and once one starts going, the other go, but you can have mutation until you are blue in the face. It's only when the structure goes that you're going to be in trouble, and the structure can go any way from outside to inside to the middle or whatever. It can go both ways. And I was just going to show you this. Did you know that if you took a human breast cell from an adult and put it in this three-dimensional gel, that's what they do, and that's how the cell is able in the breast to remodel itself again and again. We found this movement is a completely unknown movement. Without that movement, the breast wouldn't get formed. And if I had to show you the difference here, Nelly, I'm just there. Here is the normal. Look at the way it's doing it. 2 cells become 2 and they are attached. 2 become 3 and they are attached, just like a stem cell. And here are the tumor cells. And you see they have lost attachments. And if we do any of those adhesion or the polarity molecules, here are the control and here are the ones that where we remove the part 3. And if I were to do it the same thing with E-cadherin, again, you look at the movement when you have it and look at that movement when you don't have it. So every one of your tissues within a given gelatinous embryo have to do these things to make you who you are, but then they have to maintain it. And that maintenance is really the $64,000 question. So I finish by saying, here is the water coming through the sand, and it was taken from a NASA satellite. And here is the coral, and here is a mammary gland. So why is it that these forms form themselves over and over again? Fractile and the rest of it, yes, but we would need another whole meeting, as people keep saying, to discuss that. But I would say to you that we know everything about human genome, sequence of the genome, the language of the genome, the words of the genome, and we are still learning. Dictionary getting bigger and bigger, but we know very little about the language of form. And I like to submit to you that we need to learn a lot more about the spatial changes that occur to allow a tumor become tissue-specific, organ-specific, and do what it does. And so Herb Epstein is not here. I put my hand up to have a bet with him. So he, at least with regard to breast cancer, he's wrong. So again, Ole Peterson did this gorgeous paper with us, and it's really his work more than anything else. Oops, I already gave the thing to my group. But here, he did a beautiful job with 60 primary tumors, a number of cell lines. He chose a basal marker that is very tiny and doesn't overlap with a luminal marker. And then both of them are stem cell markers. And then he showed that there is indeed a differentiation hierarchy. But if you take the basal cells, they go to the luminal. Let's say A goes to B, and then B does not go to A. And so it says, yes, in the beginning there is a hierarchy. But if you take A, put it in the animal, it makes tumor. You get B, it's even more tumorigenic. And if you take all the fat cells in between, they're all tumorigenic. There is no tumor hierarchy, all right? And I don't know how to explain this, but the data are black and white, all right? So, and I just wanted to show you my very smart physicist. Not that all physicists are smart, but this one was. And she did that beautiful movement. I think I gave the rest of them the credit. Thank you very much. Lawrence Berkeley Lab. I have to tell you, Lawrence Berkeley Lab is not Lawrence Livermore. I heard last night that somebody mixed us up, and that's it. Thank you very much.